Welcome back to the second half of the Human Genome lecture. We're starting here right where we left off. I had mentioned that chromosome 1 has the most genes in the human genome. Which chromosome do you think has the fewest? It's also shown here. Well, it turns out that if we skip down here, the chromosome 1 has the largest number of genes with 2,968, and the Y chromosome has the fewest with only 231 genes. It's by far the smallest chromosome as far as information content and actual size is concerned. The way that the genes are arranged on all chromosomes is relatively interesting because the genes appear to be concentrated in little islands, so they're all grouped together, and then in between these islands there are large expanses of non-coding DNA, perhaps coding, perhaps control regions, or just perhaps structural DNA that has arisen during evolution. Here's a low-resolution snapshot of human chromosome number 1. One thing to point out here is on the left it shows you the chromosome as a cartoon with the positions given on the far left, and then there's an idea of the density of genes in the second column, which shows the number of genes in that particular location. And then on the far right are some example genes. They've been picked out randomly from top to bottom of the chromosome, just to give you an idea of some of the genes that are located on this chromosome. One thing to realize here is the genes are relatively evenly dispersed, with clumped together in these areas with non-coding regions in the middle. Notice in the centromere, in the very middle of the chromosome, there are no genes at all. That one region there has all non-coding DNA. Compare this then to the human Y chromosome. There are much fewer genes, first of all, as we mentioned, 230 or so, and they are located mostly towards the top. The P arm of the chromosome, the small arm of the chromosome, has the most genes, and then the Q arm at the bottom has very few genes. There's a whole area there with no genes at all. Another thing to point out here is that none of these genes are essential for life, because if you are a female, you have two X chromosomes and you don't have a Y chromosome. So these genes are not essential genes. But some of the examples are given here of genes that have been found on the Y chromosome. Other interesting things about the human chromosome is these large non-coding regions of our genome often has repeat DNA. So it's been estimated that 50% of our DNA is repeat DNA, and here's an example of some repeat DNA that's actually within a gene called the huntingtin gene. This is the gene that when there's an expansion of this CAG repeat, it causes the neurological disease, Huntington's disease. Now, there's a little snapshot of the huntingtin gene, and I've drawn a little box around it to sort of focus your attention. That area has the repeat, which is simply the nucleotide C, A, and G repeated over and over and over again. We all have these repeats, but people that do not have Huntington's disease have a fewer number of repeats, like 30 or less repeats. What happens in people that have Huntington's disease is that these repeats begin to expand. And so rather than having CAG repeated 30 times, some individuals have it repeated 50, 60, 70, 80 times. And when the repeat gets bigger and bigger and bigger, it leads to the expression of the Huntington's disease. On the left here, this is a diagram showing the presence of these repeats in 6 individual people. You can see the repeats from each individual. So one lane would be one person. So the first person shown there in the middle has on one chromosome 18 CAG repeats, but on the other one it looks like 60 repeats. And so this person would probably express the Huntington's disease. People that do not have Huntington's disease would not have the higher number of repeats shown in the diagram here. The Human Genome Project also has taught us about our origins. This has been an area of interest for a long time, and it's studied by scientists, biologists, paleontologists, also by sociologists and people interested in humans and how we interact with each other. Um, the archaeological evidence has suggested that human civilization first arose in Africa, and this is strongly confirmed by data from the Human Genome Project. If you look at the world and you look at our DNA, both genomic and mitochondrial DNA, it suggests that Homo sapiens originated about 200,000 years ago or so, and then all of the people that are currently on Earth came from a single large tribe of people that existed in sub-Saharan Africa, um, shown here on the map, just in northern Tanzania and um, near Somalia. That large group of people there, about 10,000 or so people, and then they, over the years, migrated out of Africa and through the world. And you can trace this by um, sequencing mitochondrial genome and also looking at the movement of our genomic DNA, particularly the Y chromosome, is used for that purpose. So the National Geographic Society has a very interesting project called the Genographic Project, and if you follow this link down here on the bottom right of the lecture, it will take you to the web page shown here. So here's the Genographic Project of the National Geographic Society. It was launched in 2005, and for a fee, I think it's around $100 or so, they'll send you a kit, and you swab your cheek cells. I'm just scrolling down here to show you the web page. I'll let you watch the movies on your own. But you swab your cheek cells, you send it back to them. They will assign you to groups of people that have migrated throughout the world. So it looks like they've had 673,962 participants from over 140 countries participate. And this is trying to develop the entire evolutionary tree of the human family, which I think is a really cool idea. I myself haven't done it, and I need to do it. So I'd be interested to see how my ancestors moved around the world and what's hidden there in the DNA. It doesn't identify any disease genes or anything like that. It just assigns you to um, a large group of people to give you an idea of your long-term history, family history on the Earth. So I think that might be pretty cool. Okay, so back to our lecture. You can explore the Genographic Project on your own. Some of what's been learned through this kind of information shows us how the Earth was populated, and a lot of this information has been confirmed by archaeological studies as well. But DNA has allowed us to clear up some certain mysteries. Like, for example, North America, it was not known how North America was populated, and there were two different ideas. One is that the original people from North America came from Asia through Russia and across the Bering Straits into Alaska. But there are also hypotheses that people migrated from the South Pacific using boats coming across the Pacific Ocean and landing along the coast of either North America or South America, and then populating the rest of the American continent, North and South America continents. Turns out then that DNA evidence shows us that the original peoples from North America came about 15 to 30,000 years ago, and they came through Alaska and then down along the coast and then spread out through the Americas. So those are some interesting things that have been learned. Other interesting things that you can see on this map here is the time scale. Now, certain places were only populated a few thousand years ago, like Iceland. You can see Iceland 1,150 years ago, populated by people from Scandinavia. Or if you look down in Malaysia, the Philippines, and these islands in the south, some of these islands in the South Pacific were only populated a few thousand years ago, and so during relatively modern history, which is pretty interesting. Now, aligned with that, as at the time that humans were moving out of Africa or migrating through the Middle East and other places in the world, we also lived on Earth at the same time as another hominid species, the Neanderthals. The Neanderthal genome has a lot of it has been sequenced, and here's some information from this one paper that I found pretty interesting. It's sort of old now, but it was the first example of Neanderthal genome, and it was compared then to the human genome. The picture there so shows this cave in Eastern Europe where they found some samples and were able to get some DNA sequence from fossils. So here's a couple of interesting comparisons between humans and Neanderthals. Did we share a common ancestor? The answer is yes. It is thought that we did share a common ancestor with Neanderthals, and it also is thought that we lived on the Earth at the same time as Neanderthals. I think modern humans, Homo sapiens, ended up being bad news for the Neanderthals. The relationship and how we arose as humans is not a linear relationship. There were several different groups of hominids that originated, and not all of them persisted. So we come from a branch that initiated, or at least one of our ancestors is this one called Homo heidelbergensis, from a fossil found in Heidelberg, Germany, but you can see the other hominids shown on here that these no longer exist. Okay, now back to the Neanderthal paper. Neanderthal fossils have been found around the world. Here's an example of one found in Israel. Imagine extracting DNA from something like this. So this is fossilized bone. They have to drill into the bone. First, they have to clean it because there's microbes and other bacteria, fungi, other kinds of organisms living on this. It has to be cleaned. Then you have to drill down into the bone to the marrow and try to extract from this rock substance DNA. They were actually able to do that and analyze these fossils found in several different locations. They took 75 samples from Neanderthal bone and teeth, and from those 75, 6 contained enough intact DNA that they could get DNA sequence, and of those 6, only 2 yielded DNA fragments that were predominantly Neanderthal. And they determined this by comparing it to modern human DNA. There were similarities, obviously, but differences. And so the 2 samples that they were able to use to get useful information was one called VI80 that came from Eastern Europe, and then El Sidron from Israel. What they found when they looked at the DNA sequences that they were able to obtain from this project, the overwhelming majority of DNA that they were able to sequence had no correlation in the existing database. And the human genome at this time was relatively complete. And so they found, perhaps, living in these samples, organisms that have not been characterized yet. And then a lot of the other ones, it's not too surprising, were bacteria and fungi, etc. But about 6% of the DNA was primate DNA, and of that, some of it was Neanderthal DNA. So this was a difficult project, particularly computationally, because they had to pick out that 6% from all the other sequences. How about this? This was a question that was answered in this project. Are humans more closely related to chimpanzees or to Neanderthals? Based upon DNA analysis, it says quite clearly that we are much more closely related to Neanderthals than chimpanzees. This was a question. Chimpanzees, we shared a common ancestor with Neanderthals and with chimpanzees about 6.5 million years ago. Our common ancestor with Neanderthals existed about 0.5 million years ago, so much more closely related. And this is the diagram here shows you this relationship, looking at individual bases at some particular single nucleotide polymorphism sites, different areas where you can tell the difference between humans and Neanderthals and chimpanzees. What is interesting, though, when you look at these DNA sequences, the overwhelming majority of all positions are identical. 98% are higher. The highest similarity is between human and Neanderthal. Okay. One interesting thing that was found, and this is not really explained yet, is that humans do share some positions with chimpanzees that are different than in the Neanderthals. So for those positions, we have the same DNA sequence that the chimpanzee does, and the Neanderthal has a different sequence. This could be due to random mutations or just simply ancestral DNA that did not mutate in the human line. One interesting example that was also discovered was that we share some sequence with this particular receptor called the melanocortin 1 receptor with Neanderthals. The melanocortin 1 receptor is an interesting one in humans because it correlates with human skin color. So you can see a range of different human skin colors in those pictures of people on the bottom left, from dark skin to very, very light skin. Overall, at least historically, darker skinned people were existed around or closer to the equator, whereas the lighter skinned people were found farther away from the equator, particularly in the northern hemisphere. And it's thought that what correlates with this is the existence of a mutated MC1R receptor. The melanocortin 1 receptor is involved in the signaling pathway that converts the signal from melanocyte stimulating hormone to the production of phenylalanine and eumelanin. And the phenylalanine and eumelanin are the two pigments that give humans their skin color and eye color to a large extent. Phenylalanine is red-yellow. All peoples express that. Eumelanin is brown and black, which people with darker skin express that. The really light-skinned person, like that red-haired lady shown there, would have very little eumelanin. And correspondingly, she would also have very little protection from the sun. But the eumelanin absorbs the UV light and helps protect her skin. Now, the wild type protein for the melanocortin 1 receptor is the one found in people that live near the equator with darker skin, and it can produce the signaling pathway that makes eumelanin. When this receptor is mutated, and one of the more prominent mutations is an arginine to glycine mutation at amino acid 307, the receptor then no longer signals properly, and it doesn't produce the proper amount of cyclic AMP, so it can't interact with G proteins, and therefore phenylalanine is produced at a higher proportion to eumelanin than in the wild type receptor. That's what this diagram up here is showing you. The MC1R is a G protein coupled receptor. Arginine 307 is the wild type amino acid in that eighth domain shown there in the cytoplasmic side of the protein, and when it's mutated to a glycine, it destroys or greatly reduces its activity, as shown in the graph on the right. What is interesting here is that the same mutation that's been found in humans, this ARG307 to glycine, was found in Neanderthals. So it's thought that maybe many Neanderthals were redheads, sort of like Lucy shown here. Other things that have been learned from the Human Genome Project is that all living organisms are related, that we have remarkably similar genetic structure to many, many animals, and even back to bacteria. You can see organizations, the same nucleotides, similar genes, etc. And this suggests that life evolved once and we are all descendants of that event. Also interesting is that the predicted human protein sequences are very highly conserved. So over 40% of the predicted human proteins share similarity with fruit fly or worm proteins, or frogs or mice, etc., which is astounding in my opinion, and that just simply reflects the fact that this gene evolved once, has a useful function and therefore it did not change as evolution occurred because it did its job and there was no need for it to change. You can compare our genome to other animals. What's being shown here is a comparison between the human genome and the mouse genome. These are the human chromosomes, the 22 autosomes, and then the sex chromosomes, showing both the X and the Y. If you compare this to the mouse genome, we have the same stretches of DNA, but they're located on different chromosomes. And imagine taking a pair of scissors, take the mouse genome, and you cut it up into little pieces, and then you randomly assemble it together into the human chromosomes. And that's what you would get here. So the different colors show you corresponding pieces from the mouse genome on the human genome. Finally, probably the biggest impetus for developing the Human Genome Project was its implications for human health and disease. Um, One of the big things that this allows us to do now is that we have a set of human genome to compare other genomes to. And so when individual people get their genome sequenced, they can compare it back to what's considered the standard human genome. And we can start to assemble these things that are called DNA polymorphisms, which are single nucleotide changes between individual people. Each individual person is almost identical to the other. So we're 99.9% identical to each other at the single nucleotide base, but those individual changes may be important. If they're occurring in gene coding regions, etc., they could affect how those genes function. And so it's going to be important to start understanding that, to start learning about human genetic diseases. There are lots of examples of this. I'm just going to give you one here. There's one single nucleotide polymorphism that is quite rare, but it's really important, and it affects the cytochrome P450 protein that's involved in energy metabolism and also in um, drug metabolism. And so this particular nucleotide polymorphism affects the protein. And so in a normal person, you can see on the left side the wild type cytochrome 2D6 gene has amino acids 167 through 170, which is histidine, serine, glycine, and arginine. There's a single nucleotide polymorphism, which is a G to T transition, which introduces a stop codon. And so now the protein stops at amino acid 168. This protein is not functional, which means that it can't metabolize certain drugs. And in this case, it doesn't metabolize a high blood pressure drug. This was discovered because there were certain people that were being treated for high blood pressure, and this drug, Declinax, is quite effective at treating high blood pressure, but in certain people, after a few days, it became very toxic, and these people became very, very, very sick. And the reason for this is that they don't metabolize the drug, so the concentration of the drug builds up to toxic levels, so they cannot be treated with this drug. Now you can see how useful this might be to know this. So now rather than testing, by just treating people with this drug, you can do a simple test to determine whether or not they have a functional wild type copy of this gene. If they don't, then the doctor can't use Declinax to treat their high blood pressure. Now this is where this is coming to. I think in the near future we're all going to have a personal chip, a genome medical on our medical cards. It'll be a functional chip where they can screen lots and lots of genes to look for our genotype at specific locations, and then better treat us for specific diseases or specific kinds of different human genetic problems. Or if we develop cancer, they might be able to take a sample of our cancer and use that to screen the chip and determine what genes have been mutated so that they can more logically treat our cancer. So I think that's going to be pretty cool, and we can thank the Human Genome Project for developing this technology. Then finally, the last thing that we should probably consider are the social, ethical, and legal issues that come along with the human genome. For example, once you have your little genetic chip, you may not want that information getting out to, say, insurance companies or other public avenues. That information should be kept secret and belongs to you and you alone. There are also limitations on genetic testing. One of the big things that's going to be a problem is that they'll be able to determine your genome, but there won't be any solution to the genetic disease, which I think is pretty sad. But I think that's going to be a big deal. Also, this issue here is still unresolved, which is patenting DNA sequences. Who owns the DNA sequence? Who can use it? Is it? I think it's crazy that they think they can patent human DNA sequences since we all have them. But that's happening right now, and that's still working its way through the courts around the world. Other issues are training physicians how to use this information, how to do it properly. Right now, it's somewhat of an art and not as easy as it seems to get reliable information from human genome, and so that still has to be affected. And then the final thing that everybody always talks about is human genetic engineering. Um, how do we work it so that you can make changes in the genome? If somebody has cancer, how do you get rid of that cancer gene? If somebody has a human genetic disease, how can you introduce the proper gene to fix that disease? Those kinds of things are going to be big and interesting issues. Okay, there you go. There's our introduction to the human genome. I hope you enjoyed it. There's no homework this week. I just want you guys to listen to the lecture and review it and hopefully learn something new.